How would you explain multi-omics in simple terms, and how does multi-omics testing differ from traditional testing?
So when you think about how something works, right, you have to understand it as a system. When we try to, and this is an engineering concept, this is in mechanics and manufacturing, you have to understand how the system works, right? Like there is no way you can diagnose things and get sort of very effective results if you're working on a machine, a device, this television, for instance, and you have no idea how it works. So you need to be intimate with its manufacture, with its processing, with its inputs and outputs, etc. The human body is the same way. So I'm not a physician, but when physicians go to work with the human body and practice medicine, they have to do the same thing. They have to understand what is working and how that's working. Now, the care of humans is so complex and time-consuming that we have sort of bifurcated the roles, right, in our modern system, where you have physicians who are largely practicing medicine, although there are MDs and research as well. And you have PhDs that are generating the knowledge, right, and translating this over to the clinical side, right? So that's how it works. So we have to still understand the system, and the task of implementing change within that system goes to the physicians, and understanding that system and elucidating knowledge on that system, that is our responsibility as scientists or researchers. So how do we do this? We have to understand how the system works, how it's manufactured, its inputs, its outputs, etc. And so that means that we take a look at how biology is expressed through what is known as its central dogma, this pervasive idea that dominates everything else. Even evolutionary theory is a thread of thought that stems from this idea that nuclear DNA, the central dogma of biology's expression, that nuclear DNA is translated to RNA, that RNA is then made into proteins, it's read and made into proteins fold, etc. Proteins interact with other proteins, with metabolites, with the gut microbiome, metabolites interact with the gut microbiome. And this is largely the functioning of the human body, right? At its most granular level, apart from sort of a biophysical understanding of the body. What's tenable in our current purview is that we grasp hold of this central dogma and we try to dictate every single level and deeply characterize every single level of this process. So we take a whole genome sequence, we take a large-scale RNA-seq, a clinically minded LC-MS proteomics panel, a deep-reading microbiome panel, and of course Metabolomics, which we currently offer at Therion. Blood proteins, a little bit of liver function. If it's necessary, we'll do, a colonoscopy. If it's necessary, and you have meningitis or a potential brain tumor, we'll do a spinal tap. If it's necessary, and we don't have to really live by this old practice that is predicated on old knowledge, right? So this is a new paradigm shift. We have tons of data, right? We have little bits of knowledge, right? So when I say knowledge, I really mean data, but they're different. We have tons of data, we're not translating it over, and the current model of testing, and testing informs like 70% of clinical decision making, the current model of testing is again predicated on old knowledge, and so it's insufficient, it's incomplete, and what we can do in research has now vastly outpaced what is possible in traditional diagnostics. So you go in for your cholesterol, you get some of this reading, you go your HbA1c, you got some of your anions, etc. And then you've got some liver function, right? That's your blood work essentially. That's your routine blood work, and we have the ability to bring forward whole genome sequencing with routine RNA-seq, routine proteomics, let's say, biannual microbiome screening, quarterly metabolomics screening. And there's a science to getting right, how often to test these different levels. But
would you say that the multi-omics approach is like essentially stacking the different systems in systems biology? And a system could apply to a lot of things. Like you could be a systems biologist just within the brain, right? Like you're looking at the various systems that end up producing, correct?
Sure. Sure. Oftentimes that's what happens.
Yeah.
So people become systems biologists of a receptor, of a receptor, seriously.
Right. But we're talking a little bit more high level where we're saying of human inputs, outputs, just how the human functions. So would you say that this multi-omics approach is enveloping or encompassing traditional testing, or that it's fundamentally different?
Oh, it's encompassing. It is completely ampliative, right? But what do I mean by that? It means that the panels that we have in systems biology totally obviate the need for almost all of these blood tests, right? And in fact, with research models, what we can do is now detect, even heavily diluted in blood, we can detect chemical signatures of local tumor microenvironments that are known to be, let's say, of the brain or of the liver or of the pancreas, et cetera. So we have gotten there. There's just not FDA approval, but the literature speaks for itself, and there is tons of data on reproducible, validated biomarkers, and we are just not utilizing them, right? The education isn't there for physicians. The technology to make this direct to consumer isn't currently available, and that's where we come in and fill the gaps.
Yeah, so there's a couple sides of this. So I want to get a little bit more granular and specific into the metabolism, but the first is that Multiomics is, so those are the various systems that make use. So when we say multi-omics testing, we're specifically talking about genome, transcriptome, proteome, microbiome, and metabolism. But there are other aspects you could put into that too, could you not? Or do you find those to be the most, you say central dogma a lot. Is that because you find that to be the most applicable to a person?
Right, so that's their molecular expression. So we would have a molecular profile. Now, just like things don't work out in physics at different scales with the same types of physics, right? Like Newtonian physics doesn't really work for like astral bodies way out there, right? That's why we have relativity. And relativity and all the physics of classical theory don't apply to very small things. That's why we have quantum physics, right? So we have rules and scales even with the expression of genotypes and phenotypes that are not necessarily translatable. So we need other data as well, right? Like we need the biosensor data. We need the magnesium, zinc, right? Like all of your divalent positive ions, positively charged ions. We need maybe we would like to have sonograph of your liver to understand the morphology in cases of non-alcoholic fatty liver disease. We might want to take, a lung function test. There are things that don't necessarily, everything begins at the molecular level really, but there are things that do not necessarily get impacted until much later, right? So you may have decreases in your rhodopsin metabolism, but you don't experience night blindness for many years. You could have, for instance, in the context of B12, we have excellent intrahepatic circulation.
Yeah, so it's finding dysfunction at a molecular level far before it gets into the symptomology or various things. Because we're talking theoretically. I'm not saying that we're doing diagnostics, but theoretically you could say, hey, on a molecular level, you are trending in a way that looks like you're going to develop fatty liver disease.
Right, or your lung function is going to be compromised, or you're going to run the risk of having, overactive immune responses. So these are the things that.
So this is kind of like a first step in test. This is like a general screening, but also an understanding of what is happening underneath the hood that's causing symptomology, but also what the trends might be for that person in developing symptomology.
Absolutely, because everything starts at that molecular level. As pathogenesis progresses, there comes a time where suddenly you just realize, oh, I'm not seeing well at night. Well, your rhodopsin metabolism has been severely compromised for the last 3 years, but there will come a definite cutoff where you start to say, oh my gosh, this is difficult now. You will get sudden neuropathy after 4.5 years of B12 deficiency, and that neuropathy may be largely unchangeable. And sometimes, people will receive their test results and they'll be kind of incredulous, right? They'll say, this isn't me. And it's like, yeah, it is you. It's just not you yet. And what we're seeing is this molecular response that is sensitive and predates the symptoms or the onset of pathogenesis by 5 years in many cases, on average 5 years. across, the top 10 killers of humanity, about 5 years. So that's where we're at, and we can extend that, because right now we're only doing really single omics testing, right? As we expand this, it's going to become, hey, at birth we identify that you have a few very probable trajectories. And then through routine RNA, proteomics, and metabolomics screenings, we are going to engineer the best trajectory possible.
So getting a little bit more granular into where we're at currently. So we talked about traditional tests, and you said a lot of it can be kind of archaic in the sense that we're looking at fairly limited scopes of what's going on. But can you kind of explain why those limited scopes aren't enough to get the vantage point that you're having with something like metabolomics? So we were talking about multi-omics. Right now I want to focus on the metabolomics portion. So essentially, why is this better than some of the other screenings that might generally be done? Or why is it different? Maybe not better. Maybe better isn't even the right term.
I think for some things for most things I would say it's better. For some things it's not better, right? And we have really great validated biomarkers, like for instance for diabetes, right? Hemoglobin A1C above a certain level, last 3 months of insulin metabolism is captured fairly well. We know this, okay? That this is a totally fine measure to use, and it's pretty accurate, right? But why is this better? Okay, so we have the ability to look at disease at a pathway level. Disease does not usually start with one metabolite. Usually, it does not start with one GPCR. It does not start with one gene. Those are genetic disorders. Those are diseases that one gene will have a certain outcome. That is not what the greatest contributors of human morbidity and mortality do to us. They are not one gene, a dozen gene, few dozen gene-related disorders. There are many, many genes, many, many effects at all levels of systems biology that have to happen in a very concerted way for the onset of cancer, heart disease, neurodegenerative diseases like Alzheimer's or Parkinson's. And so when we talk about this, you have to realize that the traditional panels are all kind of like scattered about at the pathway level, right? Disease doesn't happen with one issue, one protein, one receptor, et cetera, most of the time. There are rare diseases. There are diseases of inborn errors of metabolism, but not for most of the things that we are talking about when we are worried about our health and what's going to get us. And so the idea is that these diseases happen at pathway level, and traditional diagnostics do not look at a pathway level. They look at a few things here. They look at a few, if we look at the circuitry, the entire biochemical circuitry of the human metabolism, we see that when we map what they're looking at onto the circuitry, we see a little bit of here, a little bit of there, a little circle here for insulin metabolism, a little circle here for amino acid metabolism in the liver, a little tiny circle here for some lipids, and that is an incredibly narrow way to look at your health, okay? So they're not seeing as much. It's not that they're, these guys are stupid and they don't know what, they're just, they can't see as much. And so, of course, they're not able to render as much insight. And so what we are looking at with our panel, for instance, that we metabolism, we are looking at 68% of the 80 known canonized pathways in human metabolism. So 68% is a very good coverage, a panel coverage for this assay. And we are now able to see diseases who we know that they have pathway level effects, and we can characterize our customer samples against that known pathway level effect and show a metabolic overlap and produce a risk percentage for 480 diseases, rather than take this very narrow approach and say, well, you have high cholesterol. What does that mean? Am I at risk for heart disease? Or am I at risk at heart failure? Does this mean that I have a certain level of plaque deposition? What about my endothelial function? There's various aspects of cardiovascular health that cholesterol just does not tap, right? We think cholesterol, heart health. That is a very narrow view. Cholesterol is very good for many things as well. And so we are able to see this larger picture, and therefore we're able to match it to more things. Because again, those diseases do not happen at a single point. They happen at a pathway level. And you have to be able to analyze pathways in entirety to be able to see those diseases, characterize them, and report a risk percentage based on a metabolic similarity score.
I think initially when we launched this test, I had in my mind that we put it out there and has such a broader scope and so much more data than you would get off of most tests. And it's relatively frictionless, right? It's where the sample collection method is using a finger prick. So I kind of assumed that physicians would understand like how to incorporate this. But I think as we've been finding is physicians are trained, right, in their way of doing things. So how do you see the adoption of this evolving? Like, what steps do you think are going to be necessary in terms of educating physicians and getting them into the adoption phase of something like systems biology? A lot of the forward-thinking physicians are already considering systems biology and multi-omics and seeing that as the future. But I would say there's a lot of people that are still coming to it and asking, what's the difference between that test over there? What's this? And they're like fundamentally different, right? Like we're doing a profiling of 68% of the metabolism versus like 5% of the metabolism. And so I can explain that, but they still don't know the clinical applicability of that. What do you kind of see as ushering in the adoption of this? Obviously, things like education that we're doing right now, but just curious on your thoughts about that.
Yes, this is going to be somewhat of a shameless plug, but I really believe it is. But, yeah. So, of course, we're developing a practitioner curriculum for use of our specific panel in certain cases, right? That goes without saying, and that's not the shameless plug that's coming. That's something for, because as I mentioned earlier, we have two things. Like the enterprise of caring for humans is so complex that we need a group of people who are going to deliver care and a group of people who are going to deliver knowledge and research and data and you know. And so I'm in this latter group, and we stay in labs, right? And we essentially work on creating that knowledge, the bridging that knowledge gap as to what we know and what they can do, and working with them to bridge that gap. And so the curriculum is very important. Oncologists, as you mentioned, very advanced practitioners, especially in oncology, have been using metabolomics for quite some time and are quite interested in metabolomics. There are many metabolites that that can show, for instance, the Warburg effect, the patterns of metabolic change that show glutamine addiction, various immune escape metabolism mechanisms of this tumor in the microenvironment, even before it's a nascent tumor. Cellular escape mechanisms. These all give off chemical signatures, metabolic signatures that can be detected and used as early indicators of potential cancerous pathogenesis. And so, yes, I think that the sort of far and above physicians are already using it. Our physicians are slowly coming around to be given a chance to take the practitioner curriculum and increase their knowledge of how to use our particular panel. In a larger sense, I think what's happening is that precision medicine, not necessarily systems biology or the molecular omics, but precision medicine, for sure, that it will be a mainstay in the toolkit of specialists that practice precision medicine, is now becoming a formalized specialty, right? So I myself am the director of Metabolomics for the American Board of Precision Medicine, and it is a nonprofit entity that is sort of soliciting to become this body that formalizes a curriculum for physicians who want to specialize in precision medicine, and institutes a formal board exam to certify individuals in the DOs and MDs and physicians licensed to practice medicine, to certify them as practitioners of precision medicine. I think this formalized body is going to definitely be a key stakeholder in the future of how multi-omics is rolled out, how it is integrated into patient care, and ultimately how it's understood by physicians.
Yeah, I think education is going to be the number one thing as far as, like, there's already a lot of conversation happening around multi-omics and systems biology. You can see in the academia undercurrent that that's already in place, in the sense that it's moving in that direction. There's so many conversations, even just looking through YouTube on it, but they're only 1,000, 2,000 views because it's still a pretty small circle of people that are aware that this is kind of the future of medicine, and that will grow. That usually starts with the universities and academia, and then goes into the professions such as the MDs and the medical doctors, and then it'll kind of get out. But I think it's a factor of time is going to be a big part of it. And right now in the marketplace, there's so many different tests that people can do. And they don't have good distinction, but I think the market will catch up where they'll understand that a test that's only sequencing 4 genes is not a good test. But right now the market doesn't understand that. So that's why we're getting so many questions about like, well, how does this differ from, and this isn't actually what they're asking. They're asking about how does this differ from a particular company, but they're asking things like, how does this differ from a company that sequences 4 genes? And it's like, well, it's, you know, the body of data that you're collecting and the other technology that we have behind it that we're using to power this is so much more. And it's clear that the market just hasn't caught up onto the kind of different tests that they can do. And it's created this open road, essentially, for all these companies to come into the marketplace and provide any sort of lab services. And as long as they're good at marketing it, it seems to get sales. But I think, you know, as Warren Buffett says, when the water goes out, you can see who's naked. And that'll happen in some form or another, whether it's through regulations or just market awareness or who knows. That I think is coming. And I think that will be a big boost to this as well, as far as the mainstayers of developing and pushing this technology.
Absolutely. That's, yeah. And you said it perfectly. Whether it comes from education, and one brand or a few brands will probably rise as sort of the leaders of this as far as science and development and technology goes, or regulation, right? There's tons of regulation that unfortunately I don't agree with. I think it would be the easiest way for Theriome to succeed even more is if regulation came right now and said your test has to have A, B, C, and D, right? Half of the shops would really close, you know, and go home. We are far and above like the regulatory benchmarks that are being proposed, for instance, in the Valid Act. So we would have no problem, you know, just applying for and getting that certification if it came to it. It would be a very easy way to cull the field for us. But I unfortunately do not believe in the philosophical principles of the Valid Act and how restrictive it will make the development of in vitro diagnostics for various conditions that are of the most severe impact or of rare occurrence and are therefore not incentivized, you know, pharmaceutical or biotech industry firms from developing, right? So there are orphan diseases, there are rare diseases that nobody really picks up or studies because like the market isn't there, right? And so there are various concessions even the FDA makes for people producing therapies, for instance, for rare diseases. And the same should be applied for tests of rare diseases. And so or, you know, when we did have the COVID epidemic and we needed a test for this, right, we developed one very quickly, actually here at ASU's Biodesign Center for Personalized Diagnostics, which I maintain an affiliation still with. And we were able to develop a rapid COVID test from saliva in a matter of months, and to really sort of bog down the process of development with tons of sort of like needless bureaucratic bloated regulations. I don't think that's the way. I think the way is truly to educate people without hampering the rapid rate at which we are able to develop in science these days, and to educate people and practitioners, and people will, you know, slowly realize the benefits of, you know, you go to a company that does this really like outdated, you know, gut microbiome test, and then they're like, hey, guess what? Really strange. Your report says everything you need to take, we sell. Isn't that like, isn't that funny? Like everything that you need to take, it turns out we sell right here. Would you like it? And so we don't do that, right? Like we take a very deep look at lots of your metabolism. We tell you it's 68%. We're not lying and telling you this is everything that could possibly be seen. 68%. And it is far and away orders of magnitude above what you get at a regular diagnostic.
That is the number one litmus test, is do they sell the solution to the problem that they bring up, because that bias is inherently going to create issues in that system. Like you can't have...
We sell you...
Yeah.
We sell you the use of our analysis. which basically takes your metabolic profile, simulates a digital twin of your metabolism, simulates on that in vivo experiments of 1,300 interventions, iterated 1,000 times each, and it gauges a response to tailor a very personalized protocol for you. And that personalized protocol, we don't sell anything and refuse to sell anything on it. It's literally for you. No brand names are mentioned. It is for you. We're not in business partnerships with anyone. It is just, Theriome is just a platform for you to use these technologies that you otherwise may not have had access to, probably did not have access to. Use these bioinformatic and bioanalytical tools have borrowed from the literature to get answers that you probably would not have gotten otherwise. And then to take all of those results and query the larger relevant science and say, oh, science, what should I do to improve this? And so that's the idea, is to bring you this thing that you didn't have and not have any sort of nefarious motive. We're not selling your data. We're not selling you supplements. It's kind of that simple.
Are there physicians out there that you've seen that you think are, so nobody has successfully integrated the multi-omics, so the genome down to the metabolome and the microbiome, right? And how all that interfaces with each other, how all these molecules are interacting with each other, getting a deeper understanding of the pathways that are being affected, a deeper profiling on an individual so that you can understand their trajectory better, what deficiencies they have, all of that. So there's no one system as of now that I know of that can integrate all those things. Although I guess we do. It's just really expensive. We don't generally advertise it. It's like $20,000 to get like the N of 1 study that integrates all those things. The reason we don't advertise it is because it's $20,000, right? It's not very accessible. But anyway, it's like so there's not really this readily available market for multi-omics integration yet, because the technology is currently being developed. And we're doing that. That's our mission. But are there physicians out there that you see that are doing a pretty good patch job with integrating these things? Because what I want to get into is, like, if you see them as doing a relatively decent patch job to understand their patient better, is what suggestions then would you make to them to even improve upon that with the technology currently available or available within the next year?
Here's the thing. There are physicians right now, many of them. I can name a few, but I don't want to embarrass them on our podcast. But they are excellent physicians. And they are, for instance, in the Mayo Clinic. The Mayo Clinic has its own core facilities. So if you need your patient's whole genome sequence, you take a sample, you send it to them. They will sequence it and give you very fast results. Same thing when it comes to metabolites, right? They have a metabolomics core, a very good one. They have proteomics core. They can analyze your RNA, and it's usually by physician. It's always by physician order. You unless you're a patient within their hospital system, you don't have access to this. And then what they're doing is usually using their previous knowledge, right? So they'll be looking at, for instance, the insulin receptor. So they'll look at the insulin gene, any variants in the gene. They'll look at mRNA associated with insulin receptors, glucagon receptors. We just put a paper out on insulin signaling in corals, and we showed how intricate and complex this mechanism is. They'll take a look at protein. They'll look at the insulin receptor itself. They'll look at insulin levels. They'll look at metabolites related to insulin reactions, such as glucose 6-phosphate. They might even have some gut bacteria data, looking at what is being produced in the gut, etc. And they might piece this together given various articles that they've read here and there, and a narrative that they're well aware of. Again, it is difficult to provide people with all of this, in-depth study of care basis. Usually you have to use the knowledge you have, the training you have, to work on this patient. And so they will then take, okay, this patient is, abnormally gaining weight for apparently no reason. What is going on? Is this, a thyroid issue? Look at some thyroid genes, again, some thyroid-related proteins, your metabolites. They'll look at, obviously, iodine metabolism, and then they'll piece together, with, as you said, a patchwork of what could be going on. But they're not an AI that is trained on, the human pathways, and can, memorize and know everything that is being seen at once, right? And corroborate this to a pathway very quickly and find aberrations and mark them, and then find patterns and commonalities among those aberrations and come up with, interventions that may address 2 or 3 of them at once, limiting, the medication load on the liver, et cetera. We can do that. But there's still the human touch that's needed. So, we propose a system whereby you have an integrated analytical pipeline that takes all of this data and is able to make a cogent narrative out of it in minutes rather than, hours or days. And then it is reviewed for, right, it's sort of decomposed, dissected, and reviewed by a physician to see, okay, what is this decision-making process? What is the care that it's recommending? And do I approve or not? How am I going to modify it based on my own training? So we see this in even like radiology studies where they're looking at whether or not AI vision models are better at reading radiographic images than trained radiologists. What we find is, the AIs do better than the humans, right? But what does better than the AI is the AI plus human group, right? So radiographic images that were first read by the AI and checked by the human outperformed just the AI. And so we know that, and the same thing is true in chess, right? If you look at it, like the best chess players are the ones that have an AI, and they also use their own decision making at key points where they disagree. Chess masters who use AI, but they disagree on certain things. Even chess models are not really still able to beat out the best chess masters. In some cases, it's like 50-50, right? So the best game is played by the chess master who also has an AI assistant. And so that is where we're at in the state of AI across the board, is that it's good at most things. It's better than humans, or like 50-50 with humans. But when it's paired with humans, it's the best. It's the best solution we have. And so that is why your report is reviewed by PhD before it goes out. We have AIs that will go ahead and populate your scores, that will go ahead and calculate and produce a digital twinning platform or a digital twinning protocol for you. But that's reviewed by a physician or myself. If somebody is reviewing these reports. Right now, I still review most of them, but that is where we see is the best sort of use of AI is in conjunction with good old-fashioned natural intelligence.
Yeah, I think it's just a new technology, right? So to say you're going to go through a university now without Google would be ridiculous. It's like, why wouldn't you step upon that platform and utilize it and utilize it? Because you're just going to be better for it. It's just more information that you can access more quickly, as opposed to going to the library. So, how would you? So, like, let's say we have the opportunity to put this into a physician's clinic. What physicians do you think would be well suited for a metabolomics panel that is as broad as what we're offering? So I'm trying to get into some tangibles here. I'm not trying to make plugs. I'm just trying to help anybody listening understand where this currently fits in and some of the best use cases. I understand you can use it for so much. So maybe we kind of limit the scope a little bit and focus in on some examples, and then we can broaden out from there.
Sure thing. Obviously our test is used by some physicians. I mentioned oncologists who have a very keen awareness of certain metabolites, and they just want those metabolites. They don't even care about the rest of the protocol. They really just want levels of these metabolites, high, low, and they're trying to prognosticate the aggressiveness of their patient's tumor over the next 3 months, right? I think where that's a great use case, and I encourage it. But I think where it's best served is as a routine screening and monitoring tool, right? So if you're coming to us and you're like, I think I have Lyme disease, and we get Lyme disease like every day.
It's a little overdiagnosed,
yeah.
Right.
Like now it's just in vogue, right? Like it was underdiagnosed, and now it just seems like everybody and their sister has Lyme. And the thing is, they're not testing with methods that are gold standard. If you think you have something, well, there's a gold test for that thing, and you should go take that test, right? There are gold standard measurements for breast cancer. There are gold standard measurements for liver disease. These are tests that have very specialty diagnostics already suited for them. If you have had breast cancer in remission, well, maybe monitoring your breast cancer signature over time is a good way to keep an eye on potential recurrence, right? So that in that way, maybe it's good for you. Or if you know you have liver disease, or if you know you have a compromised ejection fraction of your heart. We had an individual who came and said, well, I have a compromised ejection fraction and my VO2 max is like 50%, and you said my cardiovascular score is a 6 out of 10. Well, yeah, but that's the physiology of the heart. There's various other things like, lipid metabolism of the heart, electrical conductance of the heart, endothelial function. These are all various aspects of cardiovascular health that our test is looking at. So in the aspects that you're not great, like 6 out of 10, you're kind of in the big part of normal. You're not in the upper end, you're kind of in the big part of normal. And that's because your other molecular functions are functioning fine as far as your cardiovascular health goes. Now, we still took into account his cardiovascular profile, his reported cardiovascular condition, and his digital twinning protocol, especially for that cardiovascular health domain, was taking that into consideration and was accommodating his compromised physiology. Now, that is one case. What I see this doing very well at, and what people are always happy with, are the sort of concierge physicians that use our tests a lot. They run usually like a boutique clinic, and so they'll see a limited set of patients, and they use this as a routine diagnostic tool, right? They want to take a big, broad, and in-depth look at lots of health domains, lots of metabolites, and use it as a first step, as a first assessment. And for that, I think it's excellent. We will recommend things if we see fit: liver function test, do a liver sonography, do a lung function test because of given levels of indoor air pollution-related metabolites. And we will give a first set of recommendations based on possible medication adjustments, supplementation adjustments, very specific changes in diet and lifestyle, and you can be fairly certain that those changes are a fantastic sort of first base, right? I also see it very well used in nutritionist clinics, so dietitians that we work with who run their own practice, and they want to, really, Metabolomics was very much not born out of, but immediately adopted by and used by the nutrition and biochemical nutrition fields. And so, dietitians who are looking to use this in their practice, this is an excellent fit, especially for cash pay customers. They come in, they get this incredibly in-depth, right, analysis of 126 markers mapped to tons of pathways, 70 different organs, tissues, subcellular localizations, 912 dysfunctional enzymes, 4,598 different SNPs. And what we're doing is we're providing this in-depth data and a first step saying, this is how they should change their diet. This is how they should change their sleeping routine. This is how they should change their physical exercise. This is how they should change their supplementation. This is how they should change their medication routine. This is very specific and granular foods, quantities, gram amounts, etc. And so that is a fantastic use case for our test, and where I've seen it be very successful. I, in fact, have a meeting with a dietitian who just onboarded with us, Micah, and she's looking to see, how can I get some marketing material and really start pumping this. So that is, a fantastic use case and something that gives a lot of early, quick direction that then they can follow up with other testing, or then they can modify as they see fit, as maybe necessary for that person's nutrition status given other tests as well. So it really is a good mold.
Yeah, I agree. I think that global profiling is invaluable because you're getting, like, if you say had to repair a house to make it the most valuable house to resell, so let's say the human body is like we're trying to optimize it. In this example, it's like a house that we need to resell. If you could get some sort of analysis that says, hey, the water heater needs repairing. This is the problem inside of it, that you need to fix this, you need to do this. And like, hey, we're giving you kind of the 80-20 view, and there is granularity to it too, so this isn't a perfect example. But 80-20 view of what you need to do and what steps you need to take and where the issue lies to get this to the highest value as quickly as possible. And so we're doing a similar thing with an individual when you're using it for global profiling. But the other side of it too that I think is pretty valuable is the monitoring through a metabolomic perspective, because if people are not doing well and they're working with a physician and the physician is trying to get them better, it is, I would say, very valuable to understand the undercurrents of the molecular underpinnings of this individual and understanding what pathways are still dysfunctional, what pathways are getting resolved, so that the physician can step on top of those things and move on to the next stage in their work with this individual.
100%. That's an excellent take, and that I guess it should go without saying, but let's say it anyways, right? Like, it's fundamental to your health. So even if you're suffering from, God forbid, a late stage cancer, understanding how your metabolism is being affected and trying to ameliorate it the best as we can, that's still a very important endeavor. Understanding what foods you should be eating, how should you be exercising? Is your biomechanics compromised? How can you exercise? What is the best way for you? What supplements can you take effects of chemotherapy? These are all things that aren't well established or well addressed by most of the medical establishment. And so that is something that our test also excels in. Because, listen, my early days in this company was, who's this test for? And my answer was always, well, if this test is for anybody who has a body. No, it really is, because at the end of the day, like, these are the fundamentals. And of course they can address certain specific. The other day was addressing a lack of sleep and a lack of energy that one of our customers had. And so she reported herself as feeling, more energized, having taken this very specific dosage of magnesium glycinate in a time of day that we recommended. But her Whoop REM levels also went up 45, 55%. And so these are tangible ways where that is good. That is a move in the right direction, whether you're perfectly healthy, grappling with terminal illness. So these are fundamentals to all things, and that is what our test can see and improve upon, and regularly does, right? We know we have a good success rate. People come back and retest, and the metabolism can sometimes be frustrating, because sometimes you come back and retest and we can't tell you you're doing better. Sometimes you come and test with us and we're like, you're excellent. See you in 18 months. No need to be worried for a while. Sort of metabolism is how dynamic and changing it can be.
Yeah, and I'm not trying to sound too self-promotional. I think we do a lot of information and education on our channel that isn't necessarily about the granularity of our test, but I think there's also the practicality side of it that's important. I think, I just lost my fucking train of thought.
It's okay.
I had a good point that I needed to make, though.
You want to take it from the top? I can go into the office. My laptop is telling me it's about to die.
Well, I got to get running here in 6 minutes anyway. I just want to, it was like going to tie the room together, and I lost it. The rug.
We're not, yeah, I know, the Big Lebowski rug. Yeah. So you were saying that we're not trying to be too self-promotional. We do a lot of self-promotion on our YouTube.
Well, we don't do a lot of self-promotion, and the reason is because we're just trying to provide education. But I think, whatever. I guess we just go forward without that. But it really bothers me because I know it was a good point that I needed to make. A lot of times I get, I know what I was going to say. So, like, we were talking about AI and how AI coupled with an individual is going to get you a better result. Just like when the internet came out and Google for a university student was a godsend, right? It really just helps to progress faster through the things that you need to do. And I view this test in a similar way, as that a physician can utilize it in the sense of they can couple with this to check off the box of that gene, that metabolome, and understanding what are the changes that need to be made from a nutritional perspective, from an environmental toxicity perspective, that are going to make pretty big improvements. And so we can kind of check that box off. A lot of people do supplementation by trial and error or a good story, good narrative weave through the supplementation that makes it sound really good, but not everybody needs a particular supplement based on their biology. Or for that matter, you might need a certain supplement, but you might need a variant of it, CoQ10 versus ubiquinol, like you said, magnesium versus magnesium glycinate versus all the different forms of magnesium that are available. These things are important for somebody's biology, and just operating off a trial and that are found in the metabolism. So I see this like when it's coupled with a nutritionist or a physician, that they get to check that box off and step to the next level, right? There's a lot of other things that they can be working with, a lot of other things that they can be doing. Get rid of the nutritional deficiencies, at least the big ones that we can identify, and get rid of a lot of the environmental toxicity impact.
Exactly. And hopefully one day when they come to Ethereum, we can give them everything from the genome down through the metabolism. But right now, of course, we're focused on the Metabolomics test that we have developed in our offering.
Yeah, we've talked a lot about the future of this. I mean, it is really exciting. I think the future of being at the forefront of precision medicine, being able to identify the best path for somebody, including, we're not there yet, but including drugs and interventions that are a little bit more invasive, including, do you really need to do this invasive treatment model, or can we find something over here that's less invasive and going to get you the same result? Well, when we separate the bias between the intervention and the testing out, we're a company that's positioned to actually do that. So I think the future of it is really exciting because it'll give people a pinpoint on what the problem is and what the intervention they need to do to resolve that issue.
All right.